Well, hello everyone and welcome back to our video series on the topic of homeostasis. So in the last video, we looked at how homeostatic control works and we looked at this thing called negative feedback. Now we're going to apply what we know to different examples in the body. So we're going to take a look at some examples of homeostatic control in the body. And in this video, we'll be looking in particular at the homeostatic control of blood glucose levels. Alright, so let's get going. Alright, so the homeostatic control or regulation of your blood glucose levels is a very important thing. Right, normally, the blood glucose levels in your blood are around 70 to 90 milligrams of glucose per cm cube of blood, or sometimes you'll see this written as DL, which means deciliter. Right, which basically is the same. 1 deciliter is 1 cm cube. Now, what happens if your blood glucose levels drop too low? Well, your cells would not have sufficient amount of glucose for producing energy through respiration. And if your blood glucose concentrations go too high, as is in the case of some people with diseases, you might have heard of diabetes, where people's blood glucose is too high, it might actually end up damaging structures in your body like nerves and vessels. And as we'll see, this can produce some pretty nasty outcomes. Okay, recall that in homeostatic control, there are receptors and a control center involved. Right, in the case of homeostatic control of blood glucose levels, the receptors and control center are found in the pancreas. Alright, however, the pancreas is quite large, so the receptors and control center are found in a particular part of the pancreas called the islets of Langerhans. The islets of Langerhans has a very funky name, probably because it was named by this guy called Langerhans. Well, but the islets of Langerhans are like little islands of cells in the pancreas. So here's a diagram. Remember the pancreas? Probably it sounds familiar, right? Hopefully from the topic on digestion. And in digestion, we saw that the pancreas secretes pancreatic juice containing enzymes into the small intestine. Here's the pancreas, and here's the pancreatic duct that carries the pancreatic juice into the small intestine. So let's take a close-up look at the pancreas. And we find that we can see the cells which secrete the pancreatic juice containing enzymes and alkaline fluid. Right, so this will go into the pancreatic duct toward the small intestine. Now, apart from these cells which secrete pancreatic juice, there are other cells which are grouped into islets of Langerhans. They look a bit like islands, right? So they're grouped into these islets, and ththese cells secrete hormones, right? They don't secrete enzymes, they secrete hormones. So if we take a closer look at the islet of Langerhans, this is one islet of Langerhans, right? It contains two main types of cells, alpha cells and beta cells. Alpha cells secrete the hormone glucagon, right? Glucagon into the bloodstream. So you can see that the islets of Langerhans contain capillaries, right? The hormones are secreted directly into these capillaries and carried into the bloodstream. Now, glucagon is the hormone that helps raise our blood glucose levels. Secondly, there are beta cells, and beta cells secrete the hormone insulin, which should sound familiar to you. Right? Insulin will then enter the bloodstream and help to lower blood glucose levels. Alright, so here's our diagram of homeostatic control of blood glucose level. We start with our set point, which is about 70 to 90 milligrams of blood glucose per cm cube of blood. Let's give ourselves a stimulus, eat a lot of donuts, yum yum yum. However, the stimulus is that the blood glucose increases to 120 milligrams. Wow, that's very high. Remember that the stimulus is detected by the receptor. So the islets of Langerhans will detect the blood glucose levels. So detect it as, oh, it's 120 units now, 120 units, and sends this information to the control center, which is also in the islets of Langerhans. The control center, remember what it does? It compares the current level, which is 120, with the set point, which is about 70. And it concludes, the blood glucose is far too high. So what does it do? It activates a corrective mechanism. And we'll take a look at this more on the next slide. But the end result of the corrective mechanism is to reduce and decrease the blood glucose level back to the set point. And once it's back at the set point, there's a signal sent to the receptor to shut off the corrective mechanism. So what is the corrective mechanism when the blood glucose becomes too high? Well, firstly, your beta cells will respond by secreting insulin, which is a hormone. Insulin will then be transported by the blood, so blood transports insulin to the target organs or the effectors, which will carry out a response. Okay, so the insulin is like a signal from the pancreas. The signal will move out from the pancreas through the blood to all the target organs or effectors. So what does this insulin do? Insulin will stimulate the liver and muscles, right, which are the target organs. It will stimulate the liver and muscles to convert glucose to glycogen for storage. So the liver and muscles will absorb glucose from the bloodstream and convert it into glycogen. Insulin also increases glucose absorption by increasing the permeability of the cell membrane to glucose. So liver cells and muscle cells become more permeable to glucose. Glucose will diffuse inside and they will convert the glucose into glycogen. So as a result, glucose is removed from the bloodstream, and this negative feedback process will decrease the blood glucose level and restore it to the original set point. Yay! So let's look at the reverse situation. Alright, so we have our set point of 70 to 90 milligrams of glucose per cm cube of blood. Now suppose you starve yourself, go, really hungry. Alright, the stimulus is that blood glucose decreases below the set point, 50 units. Again, the receptors in your islets of Langerhans will detect the level of blood glucose, which is 50, it's 50 now, and will send the information to the control center, also in the islets of Langerhans. Control center will compare the current level, 50, with the set point, 70, and you'll conclude, oh, it's too low, blood glucose is too low, and you'll activate a corrective mechanism. We'll look at that in the next slide. And this corrective mechanism will cause the blood glucose level to increase back to the set point. And of course, a signal is sent to the receptor, telling them that, oh, we're back to 70, so cut off and shut down the corrective mechanism. So what is the corrective mechanism when blood glucose is too low? Well, firstly, the alpha cells will secrete more glucagon, like a hormone. And of course, glucagon will be transported by the blood. Right? Blood will transport glucagon to the target organs, to the effectors that will produce the response. Right, so in this case, glucagon will stimulate the effectors, the liver and the muscles, will stimulate the liver and muscles to convert glycogen back into glucose and to release it into the bloodstream. So all the glycogen that's stored up in your livers and muscles will be released back into the bloodstream as glucose, right? And this process, which is negative feedback, will cause the blood glucose levels to be increased and restore it back to the original set point. Glucagon also reduces the absorption of glucose from the bloodstream, right? So it kind of like is the opposite hormone of insulin. Alright, so we've come to the end of the video. I'd just like you to really think about these questions before we end. Quick check. Define homeostasis. Can you do that? Can you define what is the function of a receptor and a control center? And what is the feedback mechanism normally involved in homeostatic control? And can you explain that feedback mechanism? And before you come to class, I would like you to also practice using this diagram that we have learned. And I want you to answer this question. How is osmoregulation, right? We learned about this in our topic on excretion and the kidneys. So how is osmoregulation a type of negative feedback control? In the case of osmoregulation, what is the stimulus, receptor, etc.? Right, actually the answer is on your textbook, page 246. But have a go and think about applying this cycle to osmoregulation. Alright, that's all. I'll see you back